Good morning. It's my distinct pleasure to introduce our speaker today, Dr. Isabelle Schrauwen. She's a human geneticist with a research emphasis in family-based and population-based methods to identify genomic factors or genetic factors involved in various forms of hereditary diseases. She recently has two R01s and I think one that should be funded. So going on three. So she's been incredibly successful in not only securing grant funding for her exceptional research, but also very well published with a H factor of 38 and over 4,000 citations. She recently joined the Department of Translational Neuroscience as an associate professor, but had previously got her PhD at the Center for Medical Genetics at the University of Antwerp, Belgium. And she did her postdoc there at Antwerp, and then she transferred to TGen uh, at our home base in downtown Phoenix. And then she went and joined Columbia University as an assistant professor before coming back to Phoenix and again, our Department of Translational Neuroscience. So it is my pleasure to introduce her and her talk, which is exploring the genomic dark matter and neurodevelopmental disorders. And I will turn it over to you.
Hello, everyone. Nice to be here Monday morning. And thank you so much for the introduction, Teresa. As she mentioned pretty well, I'm not new to Phoenix. I was here before where I worked at TGen since I think 2011 to 2017, where I was working with Matt Hellman. In between, I was at Baylor College of Medicine and then Columbia University, but now I am back with my family in Phoenix, Arizona. I'm very happy to be back. So today I'm going to talk about a section of my research, which is looking further into parts of the human genome that have kind of remained unexplored because of the limitations in technology. And I just wanted to touch upon how much the current standard technology is missing when you look at genomes. So let's get started. As you all know, the diagnosis of pediatric neurodevelopmental disorders is very important for disease management, counseling, and prognosis. Several parents of children with severe neurodevelopmental disorders often go on an odyssey to find what's actually happening to their child. Um, However, diagnostic testing, particularly genetic testing, needs improvement. It kind of depends on the type of disorder, but for neurodevelopmental disorders, both genome and exome sequencing have a similar diagnostic yield. The reason why there's so many cases that remain unsolved after genome and exome sequencing, there's several reasons for that. One of them is undiscovered genes. Many genes remain unknown at this moment. So that's part of my research is also very interested in finding novel genes associated with these disorders. There could be non-genetic causes. Sometimes we don't really understand the functional consequence of certain variants, so we don't know whether those variants could be pathogenic. Certain inheritance patterns are more complex than we understand. And then last, so what I'm going to touch upon today is that there's limitations in the sequencing techniques that we currently use in diagnostic settings. So how do we improve molecular or genetic diagnosis in these children? Um, so I'll go over this very quickly, but for example, first we can reanalyze existing data. We can look at other type of inheritance models. We can look at consider multiple diagnosis. Sometimes a child has actually a blended phenotype of multiple things actually going on. Um, we can focus on finding novel genes. We can improve phenotyping. Or what my lab is also very interested in is we actually look at different type of omics data, not just the genome. We look at RNA, epigenetics, and more. Because of that, we are able to understand the functional consequence of certain variants more, or sometimes even find variations that were hidden in the genome data. However, um, certain parts of the genomes are currently not investigated well, as I mentioned, with short read, the current standard technologies. So, what do I consider the genomic dark matter? Well, most of the human genome is actually made up with repetitive elements and mobile elements. And these elements have a very important role in genome evolution and structure rearrangement. Actually, there is a functional importance to these repetitive elements. We know that they are actually important also in the creation of de novo, which is novel structural variant creations, which are important in certain neurodevelopmental disorders. And what we see is that structural variants are often flanked by these repetitive sequences. So a standard genome currently is made of short reads. And you can see here in the top right corner, short reads are small pieces of DNA that are sequenced. And what we're trying to do is once we sequence a whole genome, is we're trying to map those short reads back to the whole genome. But because they are so short, we cannot the repetitive regions become very difficult to map back accurately to the human genome. And because such a big part of the human genome is actually made up by these repetitive elements, we actually cannot accurately figure out where these reads map back to the genome. And because of that, when we look at a human genome, we basically can only look at part of it. We're missing a whole part. Although it was sequenced, we cannot accurately figure out where it actually is supposed to map back to. And that's a problem when we are trying to find certain structural variants or certain type of variants. So the genomic dark matter to me is those regions in the genome that are currently not assessed well with short read technologies. These are repetitive or homologous sequences. For example, certain genes have pseudogenes that are very similar in sequence. And sometimes, if there's variations, we cannot accurately map it back to the actual gene or the pseudogene. And like the dark matter of the universe, the genomic dark matter is actually not well understood because of the technological limitations that we've had. And many of these regions likely contain clinically relevant regions that are probably involved in disease. And not just neurodevelopmental disorders, but also more complex diseases like Alzheimer's or late onset disorders. Um, there's new technologies that have been available that can assess this part of the human genome better. One of them is optical genome mapping or next generation cytogenetics. Also long read sequencing, PacBio or Oxford Nanopore before built, and then also artificial long reads. Today, I'm going to talk about the first two and mainly I'm going to start with a project I did on with optical genome mapping, and then I'll touch upon some examples of long read sequencing. So, based on the use of these new technologies, this actually suggests that about 22,000 to 27,000 structural variants per healthy human genome exists, and that's quite a lot. And most of these are actually not detected by a standard genome. Um, so And this is actually surprisingly high. For example, if you look at insertions, a type of genetic variant, they are missed 80 to 93% are actually missed by these short read whole genome sequencing, which actually, even using 13 different types of variant callers, which is actually the majority of insertions are missed by a standard genome. Inversions, 60% are missed. And most surprisingly to me, actually, is deletions. A lot of deletions are actually missed by a uh, whole genes, genome sequencing with short read methods. And this is because a lot of these variants are flanked by or structural repetitive elements, and therefore they become harder to see. So if you integrate all of these new technologies, you can actually lead to a high increase of these variant detection that we've been missing. So I'll first talk about one of the technologies I've been using, which is called optical genome mapping. It's also called next generation cytogenetics. I think they're trying to replace cytogenetics in diagnostic labs. And this is what it looks like. So as you can see, I'll try to explain a little bit what this actually means. But the long blue strands that you see are actually the backbone of DNA. And what optical genome mapping does is that you extract really high quality DNA. That means very high integrity DNA made of very long molecules where we label a certain a motif, and as the green sequence you see here is actually the motif that is labeled, and that creates a certain pattern. And this is then run through little channels, and that pattern is then recorded. So it's actually not sequencing. It's called genome mapping, next generation genome mapping. But to me, when I first saw it, it kind of looked like the Matrix. But I think it looks really nice to see. Um, so what we did is we took a selection of genomic cold cases with severe neurodevelopmental disorders. Um, these kids had a lot of testing done already. Exome sequencing, we did microarray karyotyping. These were families that had three families had multiple affected individuals, and then certain. We also we included a mix of probands and larger families. And then we performed optical genome mapping with 200x coverage, 200x genome coverage. And then we did de novo genome assembly and variant calling. So the data analysis actually works like this, is where all these molecules were read through the channels. We align them to each other. So you can see here it creates a certain pattern, so you can line them to each other. And then we align them actually to a consensus map of the human genome. In our case, it was human genome 38. The quality of the long reads was extremely high. So the average molecule was extreme 283 kilobase pairs. But we also had a good coverage of 200x. We called structural variants. With this technology, you can detect variants over 500 base pairs. And we detected for about 4,000 structural variants per individual, and the majority of these are insertions. So one of my interests is also trying to compare these novel types of technologies, and we found that at least for optical genome mapping, it was really good at catching these insertions that were mainly missed by standard genome sequencing. And then we annotated the variants with some in-house bioinformatics to filter out those variants that are common and that could possibly be damaging. Um, so I wanted to show you actually what it actually looks like when you look at the data. So as I mentioned, if you label DNA with a certain motif, you create a map. And this is, for example, the reference map. And then what we do is we kind of annotate our case map to, we align our case map to it. And this is what, for example, an insertion looks like. You see that there's a shift of the pattern in the case. Same with the deletion. You can actually see that there's a shift of the pattern here, and this particular pattern is missing. This is a duplication where you see the repeat of the pattern, and this is an inversion. And as I mentioned, particularly those insertions were very good to find with this technology. Of those 47 families, we found possible pathogenic variants in five. families. Actually, they're not possible. They were actually verified as pathogenic. I think when I made this presentation, I was still looking at that. We were able to reconstruct these variants further with long-read sequencing and Sanger sequencing. We also found unique and rare structural variants in candidate genes. And the majority of the variants that we found that were missed consisted of larger insertions, deletions, duplications, or inversions. So here are some examples of some of the kids that we were able to give a genetic diagnosis after optical genome mapping. So one of the first cases here is um, a child with profound intellectual disability, delayed walking, um, a very severe disorder, including skeletal abnormalities. And we found a heterozygous deletion in SON, a first deletion of the first exon. And we verified this as this syndrome in this case. Another case, for example, we also had a child with intellectual disability, and we identified a heterozygous deletion of this gene, and that was also verified to be Diaz-Loga syndrome. We also confirmed this with a blood test of fetal hemoglobin, which should be increased in this disorder, which was much higher than the normal value. Um, Another example I thought was interesting is this. This is a case with intellectual disability, delayed motor and speech development, um, that had a large inversion in a gene that should be loss of function that is copy neutral. So this was kind of invisible previously with other techniques. Although this gene is not really reported as a neurodevelopmental disorder gene, there have been two reports of complex variation in this gene in cases with a neurodevelopmental disorder. Although the disorders don't necessarily fit very well with this, you could see that perhaps this gene is prone to structural variants, and depending perhaps on the type of variant, you'll have a bit different profile of the disorder. But this is a glutamate receptor in the brain. I think you all know that glutamate is very important in neurotransmission in the brain. So we thought this was very interesting to see. The other techniques that we use for identifying these hidden variants in the genome is long-read sequencing. We use PacBio and Oxford Nanopore for that. So for long-read sequencing, we looked at cold cases where we did 15x coverage PacBio genomes, where we looked at structural variants and all types of variations. Unfortunately, we are still processing through this data, but we were able to solve some of these cases. I wanted to show you one example. This is a case, and I would try to go back to see why we didn't find this case previously or didn't diagnose this case previously. But this case, for example, had a frame shift exonic insertion in shank 1. It was not found with microarray, optical genome mapping, exome sequencing. There was actually low coverage in this region. The short read genome, the variant was seen here. This is the short read standard genome. The variant is seen, but it wasn't called by a couple of variant callers. And I think because it looked very disorganized if you look at it. But when we looked at PacBio long read sequencing, we were actually able to very accurately see the variant here, which is an insertion in the exon. And it was a great fit with the phenotype. Loss of function variants are associated with the spectrum of neurodevelopmental disorders. So after, I think, 10 years, this case was finally got a genetic diagnosis. I think this person is an adult now. So that was a really good story. But we're still going through this data, trying to see if there's other cases we can solve. And this is all new to us too. What's exciting to me about long read sequencing with PacBio is that they announced that the genomes are going to be a little bit more cheaper, $500 with a lower DNA input. And in one genome, we are able to look at both DNA variants, but also look at methylation differences. And for example, one of the things you could do is fiber-seq, which is also looking at 3D organization of the genome, and all of these things are just in one test. And with that information, I think we have a higher chance of finding out molecularly what's going on in these children. So I'm really excited to hopefully do this for all cases in the future. And I really think this is going to be implemented in the standard diagnostics in the future as well, especially with the prices dropping and the input dropping as well. This is another older case that I wanted to show you and how we identified a structural variant in a case after a long time. So this is a case with Duchenne muscular dystrophy. Now, based on immunohistochemistry, it was clearly Duchenne muscular dystrophy. However, regular sequencing of the gene, the DMD gene, we did not find a variant. After we looked at the RNA, we saw that there was a skipping of exon 8 to 51, but we didn't know why. So this whole part from here to here in this gene was skipped. So we knew there was some underlying structural variant that we were missing. We only were able to resolve it by doing long read sequencing. Um, because of that, we found that there is a very complex structural variant, which is an inversion. And the inversion was actually flanked by repetitive elements. And these repetitive elements probably masked this variant to be found with short read sequencing. So due to the limitations of technologies, we were not able to see this previously, but now we were able to verify that this is a an inversion with some flanking insertion deletions, but basically leading to a skipping of those exons and a loss of function. So I hope with this presentation, I showed you that the current technologies that we use for genome sequencing have a lot of limitations. When we talk about genome sequencing, we're not really performing genome sequencing because we're only able to look at half of the genome basically. So, but there are new technologies coming out that are able to visualize this genomic dark matter better and help solve these cold cases, these molecular cold cases that have been undiagnosed for a while. Um, the type of variants that we were able to detect with these technologies are mainly insertions, inversions, and smaller variants. That was particularly Bionano, the last one. And I think implementing these technologies in both research and diagnostics will help us understand the full spectrum of human of variation in human disease much better. Um, and in addition to improving molecular diagnosis in genes that we already are known to be associated with neurodevelopmental disorders, I do think this techniques will be able to identify novel genes implicated in these disorders, as certain genes may be in regions that are more prone to structural variants, and therefore we have missed them in the past. So I hope this was helpful, and I'd love to talk to you some more would be interested into collaborating with me. So thank you very much, everyone, and I'll take any questions if there are any questions.
Thank you, Isabelle.
I had a
Absolutely
question.
fascinating. Okay, go ahead.
Sorry, Teresa. This is Michael Kruger. Great presentation, Isabelle. I was curious in terms of the steps or the barriers that might exist in incorporating some of these techniques into standard laboratory practice or clinical practice. on a widespread level. What do you see those as being? I guess, for example, the reference data that we have for what's normal and what's not normal, or other things.
Yeah, there's still several steps to get there. I think one of the things is the DNA extraction methods. I think a lot of these techniques require very high intact DNA. So the first step is to update the extraction methods of the DNA according to that. And I think there are some more high throughput methods now available. I think they've invested a lot into trying to do that, including trying to do this on saliva samples. Trying to get high molecular weight DNA on saliva samples is difficult. And I know there's a lot of diagnostics happening on saliva samples. So that's the first step where I see a problem that we're trying to implement this into diagnostics. And then secondly, yes, the reference, this is always when we started genome sequencing in the beginning was the same thing. So hopefully there will be some more references or reference data sets available to compare these structural variants with. For example, Bionano has an in-house database that they have available for researchers to use, which is small, but it still allows us to be able to exclude those common variants, for example. But that is definitely also, like you mentioned, a critical need for improvement. And I do think that there are some big efforts on their way to perform longer sequencing on larger populations. So we would have a database like NOMID that will have long read sequence data in there. And the cost, of course, one of the most important things is the cost, which is and the DNA input, as I mentioned. There's a couple things still before this would be implemented in the diagnostics.
Isabel, it seems like a lot of the genes that were brought up that you're finding these issues with are autism related. Is that just coincidental, or are there is there something in the phenotype of the patients that's kind of trending towards those types of I guess genetic abnormalities?
Right. So the selection of these cases are severe neurodevelopmental disorders, and often these children have behavioral issues. You can look at this as a very severe spectrum of these cases, but yes, we often see an overlap with autism related genes. Um, I think these are just more on the severe spectrum of that. I don't think there was a particular selection though in this set for children with behavioral issues. I just think that because they were so severe, behavioral issues was what was included in this, and um, some of the genes are also involved in autism. And as you know, autism is very common. I think there's a lot of genes involved in autism, but this is a little bit more that is a little bit more complex.
Hi, Isabelle, it's Neil Friedman. Great talk. What I was curious is how are you sort of touched on it a minute ago, but how are you acquiring the samples, like case selection um, for it? And I think kind of what Teresa might have been referencing is there's sort of a bias towards a certain phenotype. I think the problem really is that the brain has a very limited capacity in terms of its phenotypic expression, and what we regard as neurocognitive slash behavioral, etc., is just a spectrum. And most complex genetic disorders, you see that on it.
Right. So these cases, well, this particular set that I talked about here with the BioNano set is with a collaborator in Finland. Um, we were actually, these cases were selected that they all have intellectual disability and a severe neurodevelopmental disorder that were negative in all the previous testing. Um, Not necessarily enriched for behavioral issues, though. But the criteria was that they were also negative in all of the previous testing. So maybe maybe this also says us a little bit about the fact that cases with um, behavioral issues may have more structural variants involved in disease, right? There is some kind of bias in the sample for sure. Um, but the bias is that they were undiagnosed for a long time. Um, and I do
Just for a
think,
generic database sample collection that somebody had.
yes, exactly.
And did you look at epilepsy at all amongst the affected cases that you have as a variable?
Right. Yes, epilepsy was very common um, in this set, but actually, unfortunately, we were not that successful in the cases with epilepsy. And that may be a different reason. I do think that epilepsy, we looked at a different set separately that with particularly with brain malformations and epilepsy. And what we found, what we think, and I think this is kind of a general idea, is that a lot of these cases probably have mosaic mutations, which mean that they are localized mutations that we can only find in the brain. But we're looking at a blood sample here, so we were unable to find those. So the diagnostic yield in those cases with brain malformations or epilepsy is quite low. Um, what we did for those cases, for example, was a different project where we looked at buccal cells. Buccal cells are ectoderm, so they're closer related to neurons in lineage, in development, to try to see if we could find some mutations there because we don't have a brain sample. Um, but that,
Well,
and yeah, that wasn't successful either in our case.
yeah, that's what I was going to ask because, obviously with epilepsy surgery, there's a lot of in terms of either malformative or sometimes even when you don't have obvious structural malformation, but we certainly have a lot of brain tissue from children from epilepsy surgery. So I was wondering if you had looked at brain, because I agree with you fully in regard to that mosaic stuff, whether or not you had looked at any like tissue sampling as opposed to just, germline kind of mutation.
I have not myself, but if you have some samples, I'd love to look at them for you.
Okay, so that's great. Thank you.
I, I have another random question. So when so a lot of these are changes in the genetics. Is there any common themes between the parents that make this more likely to happen? Like, are they aged? Are they um,
Dr.
oh,
Thomas, we couldn't hear you.
oh, okay, I'm sorry. I was asking whether or not there were any
Nope, we
similarities
still
between.
can't hear you.
I can hear her fine. I don't know if anyone else can.
Yeah, I can hear her fine as well.
Okay. No, I was just asking if there were any common factors with parents that tend to have children with these diagnoses.
Right. So I didn't look in this set because it's a small number, but there are some studies that obviously aging structural variants are more common when aging mothers, as chromosomal abnormalities become more common when mothers are aging. And then aging fathers also same thing, not structural variants but other type of variants become more common with aging fathers, but in these severe disorders what's the most common theme that you see is that these variants are de novo, so they are novel, so both mom and dad are not carriers of the mutation, and the mutation occurred de novo in the child, which is more common when parents are older. You have a higher risk of getting de novo mutations. And what you often see also is that the burden of de novo variants is higher. I think, for example, that's been shown in cases with autism, which is a little bit more complex as well.
There is a question in the chat. So good morning, Isabel. Nice talk. Great technology. Do all these structural variants ultimately affect expression at the RNA level or target genes? So should they be coupled with RNA-seq?
I have a note. Nice to see you here. Um, I think most of them do, but perhaps not always. Um, I do think you can mostly see this on RNA level. In this set, particularly that I've talked about, did not have RNA-seq data available to do that. But if RNA-seq data is available, that is always very helpful to understand what's going on. I think the one example that I showed here in this presentation, we actually saw the variant first on RNA level, and we weren't able to figure out what the genomic variant was, although, it was diagnostic to say, there's an inversion, there's an exon skipping in the RNA, and eventually we also found it on the genomic level. So if there's RNA available and the gene is expressed as well, um, it's very helpful in diagnosis. Uh,
And
no, Carrie did not tell me about the Duchenne case she figured out. I'll talk to Carrie.
are there any other questions? Okay, well, thank you so much, Isabelle. It's delightful to hear your talk and very nice feedback from everyone else. Uh, and I think if you put your email address back up there, if anyone has any additional questions, they can reach out to her.
Right. I do want to ask if you are interested to collaborate and you have a selection of cases that are undiagnosed. For example, if you have brain tissues or certain tissues that would be helpful to study, I'd love to work with you um, to do some next generation sequencing, and that I mean third generation sequencing or long read sequencing on these cases to see if we can find what's happening in these kids. So you have my email.
Well, thank you, Dr. Schrauwen, and welcome back to the Valley. We're lucky to have someone with your skill set here. So,
I'm very happy to be back, and hopefully I'll hear from some of you. I see some names in here that I recognize, so thank you all for attending.
okay.
Okay, well, bye everyone. And thank you, Teresa, for inviting me for this.
Uh, Thank you for giving it. It was fantastic. I always enjoy hearing it and learning more. So the genetics is always a little complicated, and it's always a pleasure to kind of learn how everything can come together for better diagnoses and treatment of the patients.
Right, that's what I'm trying to do. Well, I hope I can reach out to some more people here locally, but there were a lot of people in this call that I recognize, so it's fun to have them on.
Well, I hope that you get some contacts from this. It'd be excellent. Keep me posted.
Yes, well, I'll see you another time, okay?
Yes, yes,
Okay,
absolutely. Take
thank you
care.
so
All right.
much.
Yes, of course. Thank you for having me. Bye-bye.